Right. Hi, Jonah. You made it. Welcome.
Thank you for having me. It's great to be here.
Glad you could see some of the previous panel.
Likewise. Yeah.
Hi, Jung. Nice to meet. I don't think we have met in person yet. Thanks for joining on behalf of NIH. Hi, Christian. Very nice to have you come join us from Europe.
Thank you.
It's wonderful to have you here. Amy, as you might know, was not able to come, but she sent us a video, so I will play that. I'm very excited to have this funders panel because many of us are wondering how can funders coordinate actions, or maybe they can't. How can they nourish thousands of experts around the globe to deal with that really exciting new data, to create fair data, to create code that can be shared and that's modular enough so that it can be run and workflowed and pipelined. And also ultimately to create training and outreach and in-reach materials so that everybody keeps up with the fast pace of technology development, but also with all the new data sets and algorithms that become available so quickly and then are outdated very quickly also. Wonderful. Christian Desaintes, thanks so much for presenting on research by the European Commission.
Okay, thank you, Katy. It's my pleasure to present you the European support to the Human Cell Atlas. Next slide. This has been done through 6 pilot projects, which were worth approximately 30 million US dollars. And you see here the logos of all these six projects, which I will describe in a little bit more detail on the next slide. So Discover is a project for discovering the cellular landscape of the airways and lung. All these projects started in 2020 and ran for approximately for 13 months. The main objective of Discover was to establish the first draft of the Human Lung Cell Atlas, which was indeed launched in March 2022, this year. It represents really an enormous leap forward in the study of the human lung in health and disease. Right after the start of the project, there was a COVID-19, and Discover coordinated an international effort to analyze data from healthy people to understand how SARS-CoV-2 enters the cells in our body. These analyses identified the upper airways, nasal cavity, as the main site of entrance and transmission of the virus. Discover also contributed to several studies on the mechanism of disease in patients with COVID-19. So the project also created disease atlases for asthma, chronic obstructive pulmonary disease, pulmonary arterial hypertension, idiopathic pulmonary fibrosis, and COVID-19. These show the changes in how cells behave in different diseases. It also generated the first perturbation atlas for lung diseases, and this uses cells from healthy people or people with lung disease, and it monitors how cells change when stimulated with protein that signal to cells or with environmental triggers such as house dust mite, SARS-CoV-2, or respiratory syncytial virus. Finally, this project allowed to define local cellular neighborhoods in healthy lung tissues and the changes in these cells between healthy and diseased lung. So can I have the next slide? So this is ESPACE, which is the Human Cell Atlas of the pancreas. ESPACE is formed a merger of 3 prior Human Cell Atlas early pilot studies of the pancreas, which were funded by the Zuckerberg initiative. And now ESCAPE aimed to build the first version of the Human Cell Atlas of the pancreas, already with an emphasis on specific pancreatic disease. So the project has generated molecular profiles on cellular level, including transcriptomic and epigenomics data from more than 1 billion single nuclei, jointly together with the tissue proteomics, allowing to address fundamental questions of cell state and phenotype in healthy and diseased adults and during fetal development. So the project also collected the experimental evidence that in addition to insulin-producing cells, showing that glucagon-producing alpha cells and perhaps even somatostatin-producing delta cell also played an important role in diabetes-related pathology. So SPACE presented the first version of an open single-cell repository above the pancreas that describes all cell types in healthy tissues in type 2 diabetes and changes during aging. If I can have the next slide. Okay, the next project is UTER, Human Uterus Cell Atlas, which aim to create a single cell and a special reference map of the human uterus and provide insights into transcriptomic, genomic, and spatial changes of the organ across both the menstrual cycle and the lifespan. So it has provided the definition and organization of cell types in the main tissue of the uterus, meaning endometrium and myometrium. In addition to anti-uterine sample, UTER has performed single-cell characterization of endometrial samples obtained from patients with preeclampsia and patients with endometriosis, providing the first comparison between healthy and diseased endometrial tissues. So UTER managed to profile and study the cellular heterogeneity of the human tissue. It revealed the existence of previously uncharacterized cell states in the endometrium, including different epithelial population driving regeneration of the endometrium during the menstrual cycle. In addition, it has identified cells residing in the myometrium, including at least 10 cell types, of which 5 were poorly characterized in the literature so far. So samples were used for single-cell RNA-seq, for protein atlas, spatial transcriptomics, single-cell epigenomics, and whole genomic sequencing. It implemented an open source platform that enables data uploading and data query of raw data and analytical results. So this platform includes also advanced visualization tools that provide intuitive visualization of data with high dimensionality and facilitate comparative assessment. So this platform has been validated to identify cell type, state, and transcriptional fingerprints related to endometrial decidualization resistance in preeclampsia patients. So if I can have the next slide. Brain time. So molecular atlas of the brain across the human lifespan. The goal of brain time was to perform a comprehensive multi-omic analysis of the developing human midbrain and hindbrain and generate an atlas of key monoaminergic cell population in healthy adulthood, aging, and neurodegenerative disease. It also aimed to embed the cell type maps into 3D context and temporal developmental models. So the project delivered a comprehensive multi-omic atlas of the whole developing human brain with emphasis on the midbrain and hindbrain and identify key monoaminergic population. It also did a multi-omic single-cell survey of adult aging and diseased human brains, again focusing on the brainstem and monoaminergic populations. It integrated the in vivo human midbrain atlas with ex vivo, so organoids, to reveal differences and similarities and validate the fidelity of the organoids. And this is also in collaboration with the NIMH Brain Atlas. So next slide is the Hugo Deca, which is to describe the organization of the developing human gonads and to understand the molecular and cellular mechanism underlying sex determination. The project has built the first comprehensive cell atlas of the human gonads during development that will enable to understand the molecular mechanisms of sex determination by providing a healthy reference map to understand the development congenital origin of human infertility and to assess stem cells-derived gametes. The project has analyzed over 1.4 million single cells from 230 human embryos of fetuses aged between 5 to 21 weeks post-conception, and it has identified new cell types. It has dissected the molecular mechanism involved in the early specification of gonads into either an ovary or a testis. And in addition, it highlighted specific genes and proteins that can be used as markers to map these cells inside the gonads. And several of these genes could represent new biomarkers for differences of sex development. Can I have the next slide? Which is actually the last slide, which is the HCA organoid, which aims to establish an organoid cell atlas by combining single cell profiling and organoid technology. Using organoids obviously has tremendous potential for biomedical applications since they enable to assess, intervene, and manipulate processes in human organ systems that are otherwise very difficult or impossible to conduct in human beings. So the project has derived and characterized human brain and colon organoids from nearly 100 individuals. This resource is available as shareable living biobank for the colon and as precise protocols for brain organoids derivation from biobanks themselves. Profiling work on the single cell sequencing level was performed for all organoids, and for a subset of these, single cell imaging datasets were generated. So the in-depth processing and analysis of the single cell data is ongoing. And the next slide is the last slide where I would like to thank you for your attention.
Thank you so very much, Christian, for this really impressive overview of the work that has been completed, but also that's ongoing. I wonder if you would be able to answer one question right now before we go back to the next speaker. I'm curious to see how much crosstalk there is between those projects that you just presented. Are there annual meetings where everybody can exchange codes that might be usable across organ systems, also methods and tissue preservation and other methods that are useful not just in one organ, but potentially across many of the organs you showed? And also ultimately coming over to the atlas, typically an atlas is a book of maps, and if every single map has a different reference system, it makes it very hard to actually read and use that atlas. So ideally there would be a unifying atlas system, and I wonder if how you would encourage these kind of more global across organ initiatives while also making sure that different teams can focus. Well,
up to now, there has not been, let's say, coordination within this EU project per se, but they all interact within the Human Cell Atlas initiative, and so therefore, you know, they build some common reference with other projects, but within the Human Cell Atlas, and they participate also to the annual meetings and ad hoc meetings.
Thank you. Our next presenter is Jung Yao. He comes from the NIH Brain Initiative Cell Atlas program from the NIH. And I'm so glad you could join us today. And I could drive those slides for you, or if you prefer, you could bring them up.
Yeah, it would be great if you can run the slides.
I'm happy to do that.
Okay, great. Thank you, Katy, and organizer for inviting me. So my name is Jung Yao. I'm program officer at NIMH, National Institute of Mental Health, managing the Brain Initiative Cell Census and the Atlas program. Next slide. The research priority of this program are aligned with the Brain Initiative priorities. They recommended by the advisory committee to NIH director in their report, Brain 2025, and subsequently more recently, and bringing 2.0 to the report's recommendation. The first priority, as listed in the BRAIN 2025, is to discover this diversity, identify and provide experimental access to the different brain cell types. And the second, please just, yeah. The second priority is map at multiple scale, generate circuit diagram that vary in resolution from synapses to the whole brain. And more recently, the report of the BRAIN 2.0 highlight the human brain cell atlas as a main goal. So this is to establish a comprehensive cell atlas in the human brain that includes an anatomically informed, highly granular cell census of the whole human brain. Next slide, please. So in general, when we talk about brain cell atlas, so we are not just limited to traditional sense of atlas that's mostly defined by anatomy. And here we are talking about the brain cell atlas that's in analogy to the geographic information system that can integrate multimodal data sets across different resolutions and ontology, etc., and organize the data in a readable and disseminable way, and that people can easily access. So for brain cell atlas, we try to link data via common coordinates that we just heard from the previous panel, including reference genome coordinates and also anatomical and spatial coordinates or common coordinate frameworks, and also standardized ontology and common data elements. So the data will collect from molecular signatures that include whole genome sequencing at DNA level, and then epigenomics, transcriptomics, and proteomics, metabolomics, etc. And then anatomical features that include cell location, size, morphology, synapses, cell type composition in the tissue, etc. And also for brain tissue, as you know, the neurons, they form circuits, both at long distance projections, but also local synaptic connectivity. And then functional measures that include electrophysiology, calcium imaging, fMRI, etc. That type of data to measure the behavior and functional responses to the environment. And also clinically, and medical records and pathology type of data will be hopefully also included in this brain cell atlas data ecosystem. Next slide, please. So here is a summary of the program vision and how we organized this program. Actually started in 2014, and phase 1 3-year program mainly focused on piloting strategies and single cell methods. And 2017, we launched a Brain Initiative Cell Census Network 5-year program till this year. And so the focus is on mouse brain. We want to start small and hopefully scale fast. And so we supported a bunch of single-cell omics technology because, at that time, beginning of the program, a lot of projects use different single-cell omics approaches. At that time, the commercialization of the technology are just starting. It's not very mature yet. So we basically start with a lot of piloting different approaches, focusing on one mouse brain region, that's this mouse motor cortex, and then later joined by different teams on human and also monkey brain in the same brain regions. And now phase 3 just started. That's called Brain Intercell Atlas Network. And so next slide, please. So with a focus on human brain. So let's talk about what we have found during the BICCN phase, the second phase. So this a group of papers published last year in Nature, mainly summarizing the work in one brain region, that's primary motor cortex, applying all different kinds of single-cell omics and anatomical characterization, and some electrophysiological recording as well, and how all these data, different modalities will be mapped together. And so also the tools and resource developed, and so they can be accessible. And so this through this the BICCN network, the web portal. So moving on, next slide. So now we just founded Brain Initial Cell Atlas Network, 11 projects, 4 centers, and 4 coordinating units, and then 3 special co-laboratories. And the focus is now shifted to human brain. And so we wanted to apply the technologies validated in the BICCN in a comprehensive study, and for mainly just for human as emphasis, and try also to support multiple species and to create evolutionary, also developmental trajectories across lifespan. And so we, in addition to sequencing-based approaches, as you can see from the list of approaches we supported, a lot of spatial transcriptomics and functional as well as anatomical characterization. And as we know, currently still spatial technologies are limited in throughput. We project from these studies using current technologies, probably we can cover whole mouse brain and also several small monkey brains. And for human brain, we can cover part of brain, but not whole brain. So even though we put yearly $100 million over 5 years, but this amount of budget may still not be able to create a whole human brain about 1 liter or 3 pounds between 2 years. And so we try to support more imaging based approaches. Next slide, please. So we just published a set of RFAs, new RFAs with due date February 1st, still about 2 months to support imaging, high-throughput imaging centers. The purpose is to create comprehensive and highly granular brain cell atlas using imaging approaches. And also we want to support common imaging data processing pipeline. As you know, the imaging data currently are still quite challenging to manage in the community, and the lack of standard and unlike sequencing approaches. And then we want to support also smaller projects, R1s, to complement large scale center projects. So I will stop here if you have any questions. Thank you.
Thank you very much for this presentation. I have a quick question before we go on to the next speaker. You showed us the different phases of the projects, and from big science projects in astronomy and physics, we know that potentially these different phases require very different setups, organizational structures, leadership styles. Do you see that in the biomedical sciences also?
Well, actually, I think the phase structure for us is very helpful, especially at the beginning. We are not so certain whether the technology are robust enough to scale up. That's why we start with 3-year pilot study. And after the end of the first phase, and we are pretty confident at that time, the drop-seq, the single cell omics approaches have really evolved fast. So we seized the opportunity to scale up to mouse brain, but we are still at that time not so certain for human brain. And this is partly a cost issue for the single cell approach, and human brain is huge, and 100 million cells and 8 billion people, and so obviously we want to study not just one human brain. We try to cover the individual variation as well. In that sense, the number matters. So, yeah.
Thank you. Great answer. So I'm going to share Amy's presentation. She has sent it as a video. She was not able to attend here.
Hi, I'm Amy Bernard, Director of Life Sciences at the Kavli Foundation. I wish I could be here in person for the 24-hour Human Reference Atlas event, but I'm happy to share some thoughts about the exciting work going on with you and this project right here in this pre-recorded clip. The Kavli Foundation is a nonprofit philanthropy organization. We support basic research through our grants and endowments of scientific institutes. We're located in Los Angeles, but support science globally. I lead the activities in our nanoscience and our neuroscience programs, and I also serve as an external consultant for the NIH in their cross-divisional Human Biomolecular Atlas program, or HUBMAP. So who funds the Human Atlas? Many funders, directly and indirectly. Why? Because it's of absolute critical importance to get it right. Key aspects for the Atlas are, of course, collaboration, both from the science, but then also from the funder side. So different funding organizations might have a particular mandate to fund basic research or applied research. And so it's necessary for us all to communicate and come together on these really large critical projects. This includes things like building the community around creating the data, building community around sharing the information that's actually going to be of critical use to people to make sure that people share what's important to them. And it also means building the community in terms of making sure that participation comes from around the world, whereas really all of the different scientific areas that are required for building a strong atlas have to be included from multiple perspectives. So in my previous work with building atlases, when I was at the Allen Institute for Brain Science, I learned how important it was to have a really robust framework for any system, not just to understand its dimensions as the system itself, brain atlas, but also as a data scaffold. So this has become even more critical for a human molecular atlas, where we want to map all of the cells in the human body to a centralized space. And it means coming together in all of these different dimensions, like I said, both from the scientific side and from the funder side. So on the funder side, the Kavli Foundation covers neuroscience. We cover mostly basic science in neuroscience, basic research, and we work as part of the US Brain Initiative, as well as through our Kavli institutes around the world. So we have Kavli institutes that serve both nanoscience and neuroscience research, literally at excellent organizations globally. So the individual research projects in these different laboratories at these Kavli institutes covers questions like, how is the brain built? What are the parts? What are the structure? And all these questions really rely on a usable reference space. And that usable reference space ideally should be tied into the rest of the body. So at the Kavli Foundation, we also have grants that we do independent of the institutes. One of these grant areas that we have is in open data in neuroscience. This particular initiative supports the reuse of open data, which also greatly benefits from common high-quality reference spaces. We also support international data governance, or how different laboratories can harmonize their data from around the world as it comes down to using neuroscience-specific data in research. As part of the US Brain Initiative, we support researchers who are working on projects ranging from transcriptomics and connectivity to data integration, as well as new methods for visualizing the brain. Sculpting light, for example, and techniques for faster, higher resolution imaging. These are also critical when you think about what to map to a reference atlas. So thanks for tuning in. It's exciting to have these 24 hours to celebrate the Human Reference Atlas, and it'll be even more exciting to see the work continue that goes beyond these 24 hours.
Thank you, Amy. I think you will watch this later, and it's wonderful to have you here as part of the panel. Last but not least, we have one more speaker, Donna Cool from the Zuckerberg Foundation, and it's wonderful that you could join us today. I wonder if I should share these slides, or if you want me to share them.
I'd be happy to share my slides if that's okay.
Wonderful.
Great. Okay, so let me go ahead and take a shot at it. Hopefully it works. Can you see that?
Yes.
Now on slide. Great. So thank you very much for the invitation. I'm really happy to be here, and it's been fun to listen in on some of the prior panels and excited to also join some of the others. So I think I'll probably pick up on a number of the themes that my other co-panelists here, as well as the prior panel, shared. And I think two, hopefully, that I can really drive home. One is the importance of these atlases, and I will share with you how the Zuckerberg Initiative is supporting that, and also working with many other funders, including Yong and the Brain Initiative, and Christian and the European Commission, as well as many others, to support an international effort and this coalition of the willing that Aviv highlighted prior, to come together and bring the necessary expertise. And then I'll also try to share some of the work at the Zuckerberg Initiative that is supporting the questions around data dissemination, and Sanjay and others also highlighted this. And this is a unique feature of CZI, in which we both fund scientists around the world to do work, as well as try to build open source tools to analyze and house and share data, such that those communities, as well as the larger communities, can reference and utilize and leverage this rich data. Okay, so I lead the single cell biology program at the Zuckerberg Initiative. A very large component of this program is indeed supporting the acceleration of technologies and reference data sets that are supporting the growth of this field, and in particular, creating references via collaboration and support of these international communities generating the Human Cell Atlas. And our interest is really at this fundamental unit of life, and that's the single cell. And so much of this work to date has been focused on the transcriptomes of cells, which is the functional manifestation of the genome in cells that tells us about cellular heterogeneity. But I would also emphasize that there's a really exciting future that goes much further than that. And it includes many different technologies that are emerging that allow scientists at scale to measure diverse and unbiased features of single cells. So proteins, the genome, the spatial organization at many different scales. And this is a really exciting aspect. And then, of course, the important computational tasks of querying this data, integrating that data, and making it increasingly accessible and usable. So how has CCI supported the work to date? Much like Yong shared a moment ago, we started off 5 or 6 years ago, really focused on the methods and trying to support a set of pilots and projects that would harden those methods and ensure that when the methods were used on similar tissues across different labs, that what we were learning was not technical artifacts of an individual lab, but actually the robust biology in those samples. In addition to the data generation methods, we also spent quite a bit of time thinking about and supporting the computational tools required to analyze this data, especially now as the methods have gotten more and more efficient and the data has gotten larger and larger. I think an increasing burden on those tools has emerged. And so we continue to think a lot about that. And then what you see as we went along is that there's a series of networks here. So first the seed networks, some work on disease and inflammation, and then most recently work focused on what we've called the ancestry networks and the pediatric networks that are collaborative teams of scientists, often at least 3 to 4 groups from around the world. So we really are focused on international collaboration and supporting the growth of the networks, but also ensuring that when scientists come together, it's really leveraging the expertise needed to generate and analyze and share the data. So in the first instance, the seed networks was focused on bringing together experts on questions of individual organs, but also multiple organs. So some of the multi-organ work that Kristen highlighted, as well as Aviv, and also groups in the UK at the Sanger Genome Institute, as well as here in San Francisco at the Chan Zuckerberg Biohub. And so these groups started to establish the framework and generate those early data sets and have had an incredible impact already. What also emerged is that with those early successes are some of the gaps and some areas where there really needed to be improvement, and specifically gaps in representation of those initial data sets, both in terms of global communities and ancestral representation. And this is now an effort that we're hoping to make progress and fill those gaps so that the reference and the Human Cell Atlas is as representative as possible. And then also representation across age. So we know that these data sets are going to be incredibly important for disease research. We also know that disease does not manifest equally across age. So many diseases, including rare diseases, often arise in children. And therefore, we really wanted to focus on some of these early data sets, whereas the first instances didn't have much sampling of pediatric samples. In addition to supporting those groups, and I'll tell you a little bit more about this, we're now in a stage where to not just collect individual data sets, but put them together into increasingly comprehensive atlases requires focusing on many different computational challenges. And we've been supporting international groups to do this via a mechanism that we call data insights. So using openly available data. And Amy touched on this as well. So trying to encourage the reuse of data sets to gain greater insights, but then also building tools internally here at CCI, and specifically one called Cell by Gene that is making this data available to anyone that is interested. Okay, so this is what the CCI grantee network looks like. And I share this really to highlight just how international the collaborations are. And I think this is something that we've been very proud of and really excited about, and also maybe a unique aspect of how we can support science and help the connections of different ongoing efforts around the world come together to provide some glue to what I think has become a very large, very successful, and very diverse community that is working towards these references. In addition, I quickly mentioned Cell by Gene. This is a tool that allows scientists, and in particular allows collaborations between deeply computational scientists and maybe scientists that have less computational expertise but have deep knowledge about the tissues and cells that are being measured, to come together and to explore the differences and variation in those cells. Oftentimes by looking at different sets of genes or different features in the metadata. Sanjay mentioned some of the kidney data, so a really rich exploration of covariates, including disease or ancestry or age, et cetera. And in so doing, start to discover and in many cases annotate or share this information. Anyone can use this. I've included the link. And so you can go into the browser and open up these data sets. If you'd like to reanalyze those data sets, you can download them in whatever language you prefer and then move it into the environments. And so it's really meant to provide an open, community-driven resource that encourages the exploration and use of that data. Importantly, the data sets are now quite large. So this slide is even out of date. It accumulates really on almost a weekly basis. So there's about now 33 million cells worth of data. Much of it is distributed across a number of large organs and tissues, including the immune system, the brain. Again, I'll point to the top technologies here. So it includes many different, both RNA-based as well as other methods, and also includes data sets on different diseases and now an increasingly comprehensive number of cell types. The other thing that I'm really proud of in this session was to highlight the collaboration among funders. So as I mentioned, the Chan Zuckerberg Initiative has funded work in this space, but the way that we're aiming to build these tools and the data resources is in no way confined to the works of our grantees. And I think, as a matter of fact, the vast majority of the data that is now openly shared and accessible via Cell by Gene comes from efforts such as the Brain Initiative, Cell Census Network, and hopefully before too long, the BICAN, as well as the HubMap program, KPMP, that Sanjay included, and certainly data from Christian and the Sanger and other efforts in Europe. So it's really intended to be a resource that can be contributed to openly and then hopefully by extension used openly and easily. And with that, Bill, thank you. And hopefully we can take some questions. And again, thank you very much for joining and for the invitation, Katy.
Great overview, Jonah. And I especially like that Chan Zuckerberg also supports extensive modular software development, because that's oftentimes connected to other elements such as larger projects, but I think you very specifically support those. And for somebody who actually does a lot of tool development in data visualization, for instance, that's extremely valuable to see this kind of funding become available. I have a number of questions, and also panelists are invited, or attendees are invited to pose questions, and panelists, of course, also. You can ask each other questions. One question I had was, looking out, let's assume we have an atlas. What do you think who should host that atlas and keep it safe for the next 100 years? And of course, it will evolve over time, just like some of the human genome databases are evolving. But what do you envision as the final kind of place where it would live? Well,
I'm happy to start on this one. So I think one interesting question to think about is, what is the atlas? And certainly the data is important. And so, thinking about where the data lives. So I just shared that example of Cell by Gene, and we are certainly housing a good amount of data. On the other hand, it's a very specific type of data. So these are processed data sets. So it doesn't include much of the raw data, which currently lives in large resources such as those maintained by the European Bioinformatics Institute and the NIH and elsewhere. So I think the data may in fact live in a few places. I think where another interesting thing to think about is, as this data grows in volume, the atlas is likely to actually look more like a model, and that maybe even a set of models that drives the utility of how different scientists will want to use and ask questions of and leverage the data. And this is an exciting thing, because I think it points to a very forward-looking community and a very computationally savvy community, but also maybe raises an interesting question, because historically there haven't been places in large data repositories or traditional mechanisms to save and share and encourage the reuse of models like that. So I don't have an answer, but I think that in many ways the atlas might look a little bit different and might behave a little bit differently, which in a healthy way will push the envelope of how we think about storing it and sharing it.
Great answer, Yong.
Yeah, just I think continue what Jonah has been talking about. And so we also are very interested in this question. And but for me, I think my personal opinion is we probably should not only just think about where to host the data or store the data or manage the data, but try to think about build up some standards across different consortia. And so right now, this is a current effort of the BICAN. And so BICCN, actually, we try but haven't really got a common data processing pipeline established. And so many data generated by individual labs, processed by individual labs. So so for the ByteCAN phase, so we supported one common data processing pipeline in parallel to the individual centers projects. So hoping gradually the standards would be established, and that if that succeeds, and hopefully where data located is less of issue.
Great answer. And I know you mentioned imaging standards, and I do agree that it would be wonderful to have a Zoom event or an in-person event, preferably, that brings together leading experts and have them agree on a de facto standard, which can always be changed and improved. But I think it would be very valuable across projects to agree on an initial version of it.
Yeah.
Jonah, just to follow up, I think you're absolutely right. It's not just an inventory that we are collecting for the human body. It's really trying to get at the structure and the function. And I think the brain initiatives have been showing how that might be done. But I think there are also many other organ expert teams that are trying to get at function, not just at the parts list, if you wish. And ultimately, I think several of us are hoping to have something that you could call a digital twin that basically is born the moment you are born and then grows with you. And as new medical data sets become available, we had in the previous panel information on blood data and urine data samples that are regularly done for each one of us as part of our annual checkups. Then that gets updated and ultimately improved and can be used as a reference, as a healthy reference, because when disease strikes, something's going to change. And so you have your healthy me as a healthy reference. But this, of course, is far out so far, and we don't have that in place right now. But I think having this as a kind of future vision of how to get there and how to store and continuously update so many mini me's, if you wish, is going to be challenging, but also super exciting. And I think it would really help precision health, and also precision medicine. But ideally, we just stay healthy because we get guidance earlier, so we prevent disease. Let's see what others have to say on this argument that it might not just be a parts list, but really be a model, an AI model that we might have to store in the future.
Yeah, I don't know if Kashan wanted to comment on this. We're curious to, I think, certainly the organoid work and other models, biological models that informing this question of time resolved data or methods that are inherently destructive is a really interesting question too.
Yeah, we also try to establish a lot of animal models, not just in mouse, but also more closely to human, the monkey, non-human primates model. And so Brain Institute has actually supported another big transformative project that's called Momentarium cell tool. Basically, it's a cell tool, cell-specific targeting tools. Try to, based on the enhancers information and design the genetically targeting AAV virus and to enable specific cell type targeting, manipulating, and also recording from cell types specifically. And all the cell types underlying the circuits, brain connectivity. So hoping this kind of targeting tools can be generated through the Cell Atlas program information, the data will be used in generating these tools for developing better models rather than just computationally, but also experimentally.
And we do have a number of talks on predictive models. One is at 11 p.m. tonight, and then there are several coming from Auckland, and then also from California later in the early morning hours tomorrow morning. So please tune in for those. I think it's really interesting to see not just a kind of inventory of what anatomical structures exist and which common cell types are normally found or located in these anatomical structures and what biomarkers are currently used to characterize these cell types, but really get to the function of all of these cells. And sometimes I feel like cells are like bricks in a wall. You could actually potentially exchange them. And in fact, if one cell doesn't function properly anymore, it might get tagged, in which case it can't play anymore with everybody else. And then others take over. So it's actually a teamwork, I think, that many cells are performing in terms of functional tissue units like alveoli in the lung or glomeruli in the kidney. We had Sanjay Jain talk about those earlier today. And as part of these teams, they have specific roles to play. But if others in the team can't play their normal roles anymore, they can actually change their cell state, and in some cases even their type, to make sure that this one function, this one team is supposed to do, can be continued. And so it's, on my end, very, very interesting to see the interplay between single cells, which are obviously very, very interesting and amazing by themselves. But ultimately, they have to play in a very spatially explicit environment. And looking at that environment and understanding it deeply is also very, very valuable. And then, of course, there are oftentimes millions of these functional tissue units per organ, like in the kidney, you have a million glomeruli, and that's where the redundancy comes from. And that's if you have a tumor in part of your kidney, it can potentially be resolved by extracting that tumor, but the rest will still function. So it's interesting for me to understand what the functional tissue units in brain might be, because I believe there's similar redundancy, and ultimately to study those in addition to studying the single cells.
Yeah, I think.
Oh, sorry, go ahead, Yong, please.
Okay. So I think the cell-cell communication, I think, is pretty important aspect. So from current technologies, I hope there will be more development in that direction and to study how cell interacts and a single cell sequencing approach, limited isolation, etc. Maybe we really want to understand what spatial context, spatial transcriptomics so far, mostly just on thin sections. And hopefully, in later, so we'll also be able to study in thicker 3D environment and really see how cell interacts better. Yeah, so hopefully this technology will be developed.
And I think 3D is critically important for understand how liquids flow, so how immune cells flow through the bloodstream, etc. So we have a session at 9 a.m. on the human vasculature as a latitude and longitude system. You might be interested to join us for that one. Jonah, I think you wanted to comment.
Oh, yeah, I was going to add a quick comment that I basically just to underscore and agree that some of this predictive modeling I think is really exciting. I think it also underscores the biological relevance and the importance of the level of resolution, given that I think what we're seeing of how accurate many of these predictive models are in the early days of making critical predictions across kind of the central dogma of RNA or genomed RNA to proteins, but also across some of the other areas that Yong mentioned with species. I think the other area that's quite interesting and exciting of not just those predictive algorithms, but starting to think about opportunities for kind of synthetic information, right? So not just predicting which enhancer may be active in a given cell type, but as we learn more and more, I think we will be able to synthesize those enhancers. It may not exist naturally, but maybe exquisitely specific, which could be important for applications like gene therapy or research, as well as maybe even a future in which we might be able to synthetically generate cells. And those cells could be active for a variety of functions in cell therapies. And then if we keep kind of working up the level of scales in this discussion of tissue units, tissue engineering, and various applications, all kind of coming back to, I think, this importance of understanding that building block and the molecular definitions and wirings of it.
We have two questions from the audience. Mark Kalushka is asking, how much coordination is there between funders in the cell atlas space when planning what types of projects to fund?
I would let Jonah talk about, because Jonah is really active, trying to coordinating different funders.
Sure, yeah. So we do have what I would describe as a relatively informal mechanism of coordination and communication is probably the best way to put it. It's a regular call or gathering in which those that are actively supporting programs, so Christian and Yong and others, as well as different funders that may be interested in learning more. And I think in the past, this has been a relatively effective way of supporting this fairly distributed and fairly grassroots effort around the world. And so it includes funders from large government organizations such as NIMH and the European Commission, as well as philanthropies here in the US, as well as internationally. So it's also, I suppose, a bit of a, what was the term? The coalition of the willing.
And I guess this is especially important for big investments like scanning entire human bodies, etc., or large organs and multiple of them. Okay, there's a second question, which is Arthur Olsen. Wonderful to have you here, Arthur. And he's asking, what role has or will AI have in deep learning technologies in the use of these atlases? And I guess I would add also in the construction. I believe those atlases cannot be constructed without AI, but his question was especially on the use of the atlases.
I guess maybe imaging analysis. Image analysis, and their cell segmentation still quite challenging, and hoping AI can help in this area, and maybe also the multiplexing staining, and how AI can recognize different cell components in different tissues. In human eye, we have only 3 receptors, color receptors, but we can tell so many different things. So I hope AI can achieve this kind of capability.
Great. And I think many of us are very active on the Kaggle competitions, and that's also a way to engage thousands of teams to contribute to the creation of a open computable human reference atlas. So I would like to thank all the panelists, also Amy, for joining us on a Saturday afternoon. Thank you for that. And hopefully you talk a lot with each other to make sure that all the workshops and smaller and larger and big and humongous projects are funded well. Let us know how we can help with that all. And thanks so much for coming in and answering questions. Deeply appreciated.
Thank you very much again. Yeah, it's great to see everyone. Writing Arthur a quick answer as well, but we're out of time. Thank you.